I do have to apologize. Those in entomology can already attest to this. Explain everything went through an update today, and I'm still trying to figure it all out, but it has some really weird quirks in it now, especially with dragging stuff. It doesn't want to work like it used to. So we'll find out how long that lasts. Okay. So the citric acid cycle is a little different than what we see with glycolysis or gluconeogenesis. With glycolysis and gluconeogenesis, it was either anabolic or catabolic. Okay, the citric acid cycle is amphibolic. So it does both. To review, was glycolysis a catabolic pathway or an anabolic pathway? Catabolic, because catabolism is breaking it down. Anabolism would be putting it together, and so gluconeogenesis would be anabolic. Okay. And then, if we go, and you're going to see this a lot from here on out. Oh, that's good. We have our cell. And I'm just putting the cell behind the citric acid cycle thing. Where does okay, and then we've got, the nucleus. Yay. We've got the mitochondria. This is not drawn to scale. And don't hate me for my artistic ability, but this is definitely not drawn to scale. Mitochondria are really small. Okay. Where does glycolysis occur? Pardon? Glycolysis? Cytoplasm. Okay. What about gluconeogenesis? Where does it occur? Pardon? Part of it does. Part of it occurs, and that's why it's kind of a trick question. It kind of looks like this or this, and I'm just going to call it G-neo, because remember, it depends on what was needed, where the NADH was, what you're starting off with, that kind of stuff. So I call it G-neo because it gets really cumbersome to keep running on gluconeogenesis. But part of it, or some of it, yeah, I've already done it that way. I just made it look there. Okay, but yeah, some of it can or does occur in the mitochondrial matrix. Some of it occurs in cytosol. Glycolysis only occurs in the cytosol. Where does the citric acid cycle occur? I'll put it in a different color so that way. Where does the citric acid cycle occur? What did you say about the mitochondrial matrix?
In the inner portion.
In the inner portion of the mitochondrial. So wait. I'm confused now. So where does it occur? Let's try to just. It's inside the mitochondria. What part of the mitochondria? And it's circular, and so it's easier to draw than gluconeogenesis here. What part of the mitochondria does it occur? Yes, asterisk. Okay, so it looks something like this. So Quentin, you were right on both accounts. So that's the citric acid cycle. I should have made it bigger, sorry. Okay, and the reason why is we're going to find out that one of the enzymes in the citric acid cycle is actually stuck in the inner membrane of the mitochondria. And it's also shared with another metabolic pathway. Does anyone know already what metabolic pathway shares an enzyme with the citric acid cycle? And that metabolic pathway occurs in mitochondrial inner membrane. I'm sorry. It's an electron transport chain. But yeah, that's why I kind of do it like that. 7 of the 8 enzymes, not including what I call step 0, occurs in the mitochondrial matrix. Mitochondrial matrix is the holy of holies of the mitochondria, so to speak, for phase integration. And then we'll find out that, and I won't give it away, but one of them does occur in, it's a part of a large complex of the electron transport chain, so it occurs, but it's stuck in the inner membrane. Okay. And so that's what this is all saying, that it occurs in the mitochondria, blah, 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 blah. And so this is like a good summary of everything. I'll give you a moment to get caught up, because it actually also includes some stuff that we haven't covered yet. But I like this came from the old book. Because what's the overall goal for like glycolysis and citric acid cycle and all that kind of stuff? Well, ultimately you're going to make ATP, but do you make very much ATP directly from either one of those? No. What do you make that's even more important? It's NADH or FADH2. So that's what they're trying to show here with the little electrons that are floating. If you remember from organic chemistry, you don't just have electrons like pop off like that, usually. So you're going to actually find that there are ways to violate that rule. You know, in organic chemistry, especially too, say no, we don't just pop electrons off. Well, in vivo it does happen once in a while. That's not good. But they're showing it here. In reality, these E's, the E minuses, really are in forms of either NADH, or FADH2, or FMNH2 and ADPH. All right, but what we have here in what I like is ultimately here what they're showing is you're making this Acetyl-CoA for the most part. And you're making these, like I said, NADH or FADH2, depending upon what it is, which will ultimately go on through and be part of electron transport chain. It may not be directly, we've already talked about how the fact that an NADH that's made in the cytosol can't just pass over to the mitochondrial matrix. Okay, so there are ways around that. But acetyl-CoA is like a common intermediate here before you get to the acetyl-CoA oxidation. And the citric acid cycle also generates these reduced electron carriers, which then ultimately will go to the electron transport chain for oxidative phosphorylation. Or if you prefer to see it vertical, it's the same thing, but this one came from your current book. Horizontal, vertical, whatever. Like, this one's a little more colorful. So on your own, you can go through and tweak that. Okay, so this is the citric acid cycle. There are 8 asterisk steps to the citric acid cycle. I call that because there's one that I literally call step 0. Once in a while, some books will put it in as a part of the cycle. It's the one that's up there at the top because it's not an actual part of the citric acid cycle. And it's also a chance for me to do phase integration. Okay. So I call it step 0. Let me make this smaller. Because glycolysis ultimately made pyruvate, okay, and NADH, and 2 equivalents of ATP. But the pyruvate, the problem is pyruvate does not directly enter the citric acid cycle. Much like Moses with the Holy Land, it doesn't get to enter in the citric acid cycle. It first needs to be converted. And that conversion is where my analogy with Moses ends. But that conversion is to where we get pyruvate to acetyl-CoA. I do expect you to be able to draw what the acetyl group is, and for CoA, you can just write out CoA, because coenzyme A is really huge. We've talked about that before. All right, so this is the extent of you drawing acetyl-CoA. If I ask you for the structure, this is good. Okay. By this point in time, you should know what pyruvate looks like. You should be able to draw pyruvate. Just remember what amino acid looks like pyruvate. Alanine, and then you just substitute the amino group for the ketone, okay, because it's the keto form. Alanine is the amino form. This is called the alkyl acid of alanine, or alanine is the amino acid of pyruvate. And we'll see that over and over again. Okay, so we take a pyruvate, we take coenzyme A, which the important functional group on coenzyme A is the free thiol. The rest of coenzyme A is just, this is the business end, is what my old teacher used to call it. The rest of it's for like binding and recognition and stuff. But it doesn't do the catalytic portion. We have an NAD plus. Ultimately, you're going to get acetyl-CoA. You get carbon dioxide, and you get an NADH. And what we'll see is that each NADH could ultimately make up to 2.5 ATP, depending upon the location that it came from. And we're going to find out that coenzyme A has a lot to do in both the breakdown and the synthesis of fatty acids. Okay. So remember, this is what I call it step 0. They don't call it that. What class or classes of enzymes could this be? If it's a transferase, what would it be transferring?
The business end.
Let's give it the business end. Yeah. Okay. Oh, and someone said that CoA. Someone had said that, or you could say, well, it's transferring. What about if you're going backwards? It's transferring carbon dioxide, the carboxyl group. What else could this be? A what? It could be decarboxylase. That is true. What else could this be?
Dehydrogenase.
It could be dehydrogenase. That is also true. The answer is one of those is correct. No. This one is a little bit more complex than that. This is pyruvate dehydrogenase complex. Okay. It's truly a dehydrogenase. It is doing decarboxylation. It is, you could say, doing the coenzyme A transferring as well. This, and many times I will probably use this as PDH. This complex is huge. To quote our president, huge. But yes, it's a very, very, very large complex. And it has several cofactors, okay, since so much goes on for it to occur. It's got several cofactors. Some of these we've seen before, some of them we may not. This is by far an oversimplification. I actually don't like this figure because I think it takes something and makes it way too simple. So you get pyruvate from glycolysis, you get acetyl-CoA. One of the other places acetyl-CoA is beta oxidation. So what is beta oxidation? What is beta oxidation?
It's saying it's on beta carbon.
It is. It's when we see beta oxidation of fatty acids, that's the breakdown of fatty acids. That's the metabolic pathway that breaks it down. Okay. This is the pyruvate dehydrogenase complex. You can see already. Well, one thing it's going to require is NAD plus NADH. Another thing it's going to require is coenzyme A. But it actually requires, like I said, a lot more. I like this figure better. I'll give you a moment to write it down. It requires five different things. And that's not even to say magnesium, which I'm sure is floating around as well. Since you can't have a carboxylate without a magnesium. We're going to break it down, go into more details here, but I just want to give you a moment to write it all down. The five are NAD plus NADH. Remember, that's the two that go together. Coenzyme A, which becomes acetyl-CoA. TPP, which we've actually seen once before. It's been a little while. We'll talk about it. Lipoate, which is also called what? Where do we get lipoate? It's lipoic acid. It's something that you need. And then FAD, which seems really counterintuitive. Okay. Because you do not get an FADH2 out of this. You get an NADH. And I'll explain how that happens in just a moment. Technically, there are 3 enzymes that come together to make the pyruvate dehydrogenase complex. Does anybody, maybe this, at least back when my brother was a kid, he's still considerably older than you, there was a cartoon that's called Voltron, or even, what's the one that was like, Planet? No, Captain Planet, or whatever, I can't think of it now. Maybe I'm gonna have to look it up. But Voltron is where they kind of look like these cats, but then they come together and they form like this really big monster kind of thing, and they would all come together, that kind of stuff. This is kind of like what happens here, because we have 3 different enzyme functionalities, which you were saying most of these functionalities, But they all have to come together in order to make the complex, to make the enzyme, to make it do what it needs to be done. On their own, they aren't powerful, but together they are. Okay? That's what the E1, E2, E3 means. Those are the three components. Like I said, we'll break it down in just a moment. So CoAsh is just the way to abbreviate coenzyme A, NAD+, ADH, TPP, lipoic, okay, yeah, all right. Oh, why is it moving everything? So we have seen TPP. What did TPP do? I mean hyperphosphate. What does it do? It's been a while. TPP and lipoate, or lipoic acid, do very similar things. Okay. What they're involved with is many times it's important because it activates or helps with the transfer of acetyl or acyl groups. Okay. Acetyl is a specific one, meaning it's only got 2 carbons. Acyl doesn't define how many carbons it has. Okay. That's why many times we'll say fatty acyl groups. But this time we can specifically say acetyl because it's Acetyl-CoA that pyruvate is being turned into. This is what some of the other cofactors look like. We talked about this at least once before coenzyme A, like really early on. This is what it looks like, and that's why I said this is the business end. It's reactive thiol. When it's Acetyl-CoA, it just has, instead of the hydrogen, it makes it thioester. But this is one of the reasons why you also need pantothenic acid. This is derived from adenine, as you can see, but all of this doesn't have anything to do directly with the reactivity. It's only this thiol that's important. Here is lipoic acid. Lipoic acid gets added covalently to lysines. Okay, so normally, and the reason why it's called lipoic acid is whenever it's a free, it would be free carboxylate or carboxylic acid. But here it's been, it's got an amide connection to the lysine. This is where it's oxidized, and this is its reduced form. And as its reduced form, you can actually use these thiols to add in acyl groups. Okay. All right. This is the pyruvate dehydrogenase complex. It is definitely a textbook example, pun intended, of substrate shuttling. So each one of the E's, E1, E2, E3, they actually have a name as well, or even a functionality, if you want to think of it. The very first, and it should make sense, the very first one's called pyruvate dehydrogenase. The second one is dihydrolipoyl transacetylase. And the third one is dihydrolipoyl dehydrogenase. Okay? Give you a moment I want you to look at it, and then we'll discuss it. That way you can try to follow along a little bit better if you look through it on your own. In the beginning, we're going to start with pyruvate to ultimately talk about, making the acetyl-CoA. So we have pyruvate. The very first step is going to be the decarboxylation. The way that this works is the pyruvate dehydrogenase portion will actually take the TPP and it will be acetylated. Okay, and it's acetylated in the form, and they call it hydroxyethyl. Plus you see ethyl, there are 2 carbons, and then the hydroxy group. We'll see that later on. I'm sure you'll probably forget that in the future as well. And then we remove the carbon dioxide. But we got to regenerate it. We haven't made acetyl-CoA yet. So then the second portion of this complex is the dihydrolipoyl transacetylase. So that tells you it's going to be transferring an acetyl group, and it's transferring the acetyl group to a dihydrolipoyl group. Okay, this is a mouthful, but this is the oxidized form, and this is the reduced form of the lipoic acid. And if you remember back to the rules, I didn't teach you organic chemistry 2, but when you take a carboxylic acid and you modify it, remember you drop the ic acid and you add whatever, and that's where you get this oil. OYL means it means it's literally the lysine is attached to the lipoic acid. Okay, I don't know if you guys remember that or not. This is where it looks really weird, but if you break it down, it makes perfectly logical sense. The lysine that's attached to lipoic acid. This is the oxidized form. This is the reduced form. And so now the acetyl group gets transferred to it, and in the meantime, the TPP is regenerated for the next round. So now we have the acetyl group on the lipoate, and this is where coenzyme A comes in. Coenzyme A will come and knock it off in order to regenerate, well, not regenerate it yet, but in order to make the reduced lipoic acid. And we have our acetyl-CoA. But this can't be the end of the story, because otherwise we have SHs here, and we need it to look like this. That's where the FAD comes into play. So then the last dihydrolipoyl dehydrogenase, this is a dihydrolipoyl group. What it does now is it's going to take the FAD, turn it into FADH2. So it takes the electrons from the SH in order to make the FADH2. So now we're back to the lipoic acid being in the oxidized form. But the FADH2 needs to be regenerated to FAD. And that's where the NADH comes in. And the NAD plus becomes NADH to regenerate the FAD for it to repeat the entire process. So it's like little gears. You want to think of it that way. And you ultimately make NADH. And once again, NADH, depending upon its location, I think can go up to make at least, or make, I should say at least, can go up to make at most 2.5 moles of ATP. If you just left it with FADH2, it could only make 1.5 moles of ATP. Okay. And I do suggest on your own that kind of work the logic on the naming and like what all is going on here, because it is busy, but it's very important. And we're going to see something like this soon. We're going to see this again, this kind of thing. It is huge. And to me, it looks like honeycomb cereal. I don't know why. I don't know, it just reminds me of honeycomb cereal. But you can actually see the E1, E2, E3, they've got them color-coded here. So it's like it's an assembly factory. It's just pumping, taking those pyruvate and pumping out the acetyl-CoA and the NADH. Okay, so it just passes it along like a little assembly line. And once again, it's huge enough that you can actually see it using microscopy, which we can't see that a lot of times with enzymes. Most of the times enzymes are small. Too small to be able to differentiate using just something as simple as microscopy. But you can see the little honeycomb kind of things. Maybe it's just that usually about this time I teach biochemistry and getting hungry. Okay. Are there any questions so far? All we've done is what I call step 0. And from the products of step 0, we actually make an NADH and acetyl-CoA. The acetyl-CoA is what's going to go on to the citric acid cycle. Any questions? This is a citric acid cycle. Once again, when you get a chance to look at it, you just see the types of reactions. We're going to actually break it down and go through it. And by the way, the citric acid cycle with the blanks is already available on MyFire. So, pyruvate dehydrogenase complex was step 0. Now we're in officially step 1. We have acetyl-CoA. Acetyl-CoA is going to hook up with oxaloacetate, which we had talked about once before. Where have we seen oxaloacetate? It's part of gluconeogenesis. We can also get oxalic acetate from one other place. So it's in the citric acid cycle, it's in gluconeogenesis, and it's in one other way, at least so far, that we've actually talked about briefly in the...
Aspartame?
It's aspartate. Mm-hmm. Because this is the alpha keto acid version of the aspartate. You turn that ketone into an amino group, you've got aspartate. And it's aspartate aminotransferase that does that. So oxaloacetate is another one of those common metabolites. Or if you've ever been to someplace like, Paris or DC or something, you've done the subway system. They have the common termini. It'd be like a common terminus. So, and a lot of times we abbreviate, or at least I do. I think your book does too. Oxaloacetate is OAA, especially if you write it over and over and over again. But if we have OAA and acetyl-CoA plus water, we make citrate, which citrate, if we think back to Sesame Street, has 1, 2, 3 carboxylic acids. Kind of thing. So that's why it also is called the tricarboxylic acid cycle, citric acid cycle. And we get the coenzyme payback, which can then go on to do this process again. All right. What class or classes of enzymes could this be? Think about it. What's one of the possible classes of enzymes? And if you need to, start going through them in your mind. Is this a cyclase? No. Okay, is this a dehydrogenase? What's missing if it was a dehydrogenase? NAD, NADH, FAD, FADH2. So the answer is no. Okay, so what could this be? Acetyltransferase? It possibly could be an acetyltransferase. All right. What else could it be? If it was named for the backwards reaction. What else could it be? What are some of the other classes of enzymes? Is this a kinase or anything to do with the phosphates? No. There's at least one class that you're forgetting all about. What? Synthase. Because what do synthases do? They synthesize something. What are we doing here? We're synthesizing something. What are we synthesizing? Citrate. Okay, and so then we have to say, okay, it's either going to be a synthase or synthetase. Which one is it? Synthase. Why? We're not using up ATP or GTP. So this is citrate synthase. Oh, no. Didn't mean to do that. Yeah. So yes, this is citrate synthase. So step one is citrate synthase. It is an allosteric enzyme. What's one other note that you can make about it? State the obvious here. What is it or what can it not do? It's not reversible. So therefore, it's one of the checkpoints. Technically speaking, there are 3 checkpoints within the circle. 4 if you count step 0. So that's why you have to really look. Either if this was an essay question, you'd have to be very specific and explain it, or look to see what the question is reading, because you're going to do that a lot, unfortunately, with the citric acid cycle, because it's got that little step 0. But within the circle, there are only 3 checkpoints. If you include step 0, there's 4. Okay. This is 1. Okay. Regardless of what I'm doing. So it's an allosteric enzyme. What are some of the things that you would expect? Are you going to want to do citric acid cycle whenever you have low energy or high energy? Low. So then, what's an example that you would have if you have a high energy? High ATP. ATP inhibits it. What's another one?
Low ATP.
Well, for inhibitors. Besides ATP, what's another?
NADH.
NADH will inhibit it. And then there's a third one, which I wouldn't expect you to know at this point, and that's Acetyl-CoA. Does anyone know where Acetyl-CoA comes in at? It's derived from succinate, but it's something further on down in the cycle, and so that's why it should make sense. Feedback inhibition. But yeah, these first two should make perfectly logical sense at this point in time. You have a lot of NADH, you don't need to make more. Right? You have a lot of ATP, you don't need to make more. And if you have a lot of Acetyl-CoA, that means it's already backing up later on in the cycle, so therefore you don't need to make more. This is just a little picture showing all of this and how it comes together. It's huge, once again. Okay. Then we have step 2. Remember, there are only 8 steps, not including step 0. This is step 2. Oh, every single semester, I forget to wear my t-shirt, even though we don't wear t-shirts very often. I've got some t-shirt. This is called a Ferris wheel. Does anyone know why? It's the Fe2+. That's the ferrous ion. Ferrous ion versus ferric ion. Yeah, it's called the Ferris wheel. It'll make sense once we talk about it. This one's very different for us in the sense that it uses iron. Okay, so that's very important. This is also one of the important steps. In the sense, it is not a checkpoint, even though it's irreversible. Now I'll talk about the reason why. It is not considered a checkpoint. This is important in the sense that if you look at, hopefully the way it's drawn, it's a little bit more obvious. Citrate is achiral because this central carbon has 2 identical things hanging off of it. But it takes something that's achiral and turns it into something that's chiral, which you don't usually see that. And if you remember back to organic chemistry, one of the pitfalls for organic chemistry in synthesis in vitro is that usually if you start with achiral reactants, you're going to end up with achiral products unless you can somehow force it into a chiral environment. Our body does that naturally with enzymes. Okay, so a lot of times they even use this enzyme as an example of that. Another thing is this is technically, this step, sometimes they write it as step 2A and 2B. It's the same enzyme, and it makes an intermediate, but the intermediate never leaves. The intermediate is called a connotate. So the connotate's not shown right there. Does anyone know what the name of this enzyme is? I'd almost give you bonus points right now if you knew it. It's named after the intermediate. I don't know why. It makes no logical sense. This is one of the few that I can not explain away. It's named after the intermediate that never leaves. Okay, so in a moment I'll try to show you an example of what I mean. So what happens is it takes citrate, transforms it into a connotate, then takes the same enzyme takes the connotate and makes it into isocitrate. Then whenever they did the naming, they named it after that intermediate, which is not even shown here. And so sometimes when you see this written out, which I don't have it, and next slide, I thought I did, then it looks like it's 2 separate steps. It's really all the same enzyme. Okay, and they named it after the intermediate. This is a connotate. It's just one you have to memorize, unfortunately. This is the Ferris wheel. This is the very first time I do believe that we've seen an FES, an iron sulfur center. We're going to see these later on. Iron's very important. We've only seen iron, I think, in one other instance. What's one other instance that we've seen iron? I can think about both off my head. Last semester. Hemoglobin, and myoglobin, right? For holding on to oxygen. Well, iron sulfur centers, and I'm not an inorganic biochemist, so I can't explain it other than just to tell you it is the way it is. But these FES centers, you can have different kinds based off of the geometry. Some of them are called FES, some of them they'll say are Fe2S2. They can have up to 8 different sulfurs. But based off of their geometry, it affects their ability to transfer electrons around. And so just a little slight twinge to it can really have a drastic effect on that electron potential. So like I said, I don't know the reason why the iron and the sulfurs do that, like what makes it so special, but it's a special bond, special type of thing, and it makes that palladium ferrous yield. Okay. All right. All right. So step 0 was a pyruvate dehydrogenase complex. Step 1 was citric synthase. Step 2 is a condensase, which has the ferrous wheel. This is step 3. Just looking at it so far, what's one thing that you can point out with it? Even if I didn't uncover any of the blanks. It's a dehydrogenase. And why do we know it's a dehydrogenase? NAD and ADH, yes. What's another observation you can make, even if I don't uncover any of the blanks? It does. Is it intermediate called oxalosuccinate? Pardon?
Well, the alpha-ketoglutarate.
Alpha-ketoglutarate, it looks like. This is the first time I think that we've seen this. Sometimes they even abbreviate alpha-KG. It's very important to multiple metabolic pathways as well. It's glutamate. It's the alpha-keto acid of glutamate. This is like OAA, oxalic acetate, is aspartate. Alpha-ketoglutarate is glutamate. It's alpha-keto acid. So you should be able to show that. But also, if you notice, it's irreversible. This is the second checkpoint within the cycle. Okay? Third if you count step 0. So now that we know the class of enzyme, it is a dehydrogenase, what do you suppose you can name this one? There's only a couple names that would make sense. It is truly a dehydrogenase. You're making NADH, NADH. This is isocitrate dehydrogenase. It's a checkpoint. It's important. What would make sense that would inhibit it? What's one thing that you think that could be possibly right here? It's ATP. It's inhibited by ATP, and it's inhibited by NADH. So if it's inhibited by those, what do you suppose can activate it? It can be activated by ADP and NAD+. So see, you guys already know. Yes, it's isocitric dehydrogenase. We'll see alpha-ketoglutarate in the future again. The enzyme that transforms glutamate to alpha-ketoglutarate and vice versa is an aminotransferase. And so, trust me, by the end of the semester, alpha-ketoglutarate you're probably going to get tired of. I shouldn't say. It's one that you see a lot. This is just the first instance of it. Okay, and I think this is a good stopping point. So now we'll pick up where we left off. So now we're up to step 4, which is where we have alpha-ketoglutarate. Oh, one thing we did talk about, alpha-ketoglutarate is the alpha-keto acid of what amino acid? Glutamate. Okay? Once again, all you do for those alpha-keto acids is if you transform the ketone to the amino group, which we'll see aminotransferases, that's what they do, then that is glutamate. Just like oxaloacetate, OAA, is the alpha-keto acid to aspartate, and pyruvate is the alpha-keto acid of what amino acid? Alanine. So that's why those are probably the 3 things that I would expect you to know their structures, just because you already know the structures of their amino acids. Okay. So we take NAD+, you get NADH. It does utilize coenzyme A, and this is where we get Acetyl-CoA. I will just tell you right now, this one is also, even though it's reversible, it is a checkpoint. It is one of the ways that they keep the cycle under control. Okay. So, what class or classes of enzymes can this be? Or should I let me do? Does it look an awful lot like anything that we've seen thus far?
Dehydrogenase.
It's a dehydrogenase. Yes. And why is it a dehydrogenase? Okay, NAD going to NADH. Yes. Does it look like anything that we have seen so far this semester? Look at what all it requires. It looks like that complex and left complex was pyruvate dehydrogenase complex, and this is very analogous to that. So knowing, now put the pieces together, so what would you call this one? What enzyme is this?
Alpha-ketoglutarate dehydrogenase complex.
Exactly. The alpha-ketoglutarate dehydrogenase complex. It's very, very, very similar to the pyruvate dehydrogenase complex, but it's a pyruvate. We're working with alpha-ketoglutarate, and we get succinyl-CoA instead of acetyl-CoA. So that's the big difference. And I would never ask you how many kilojoules per mole, all that kind of stuff, but it is. It requires the same, TPP, lipoic acid, FAD, Acetyl-CoA, and NAD. Same setup as before. Okay. Are there any questions so far? Anything? Okay, remember, and so we've actually gone over the checkpoints already. Not that step 0, pyruvate dehydrogenase complex, is kind of like a gateway. Then we had steps 1, 3, and 4, citrate synthase, isocitrate dehydrogenase, let's try because I don't have the numbers memorized together, and alpha-ketoglutarate dehydrogenase complex. So now we have succinyl-CoA, which remember succinyl-CoA is actually one of the things that can feed back and inhibit citrate synthase, or step 1. Okay. I give you a moment to look this over. We're going from succinyl-CoA to succinate. What's one observation that you can make about this? And there are lots of them, but what's one?
It's one-sided.
It is one-sided. It's essentially irreversible.
It utilizes inorganic phosphate.
It uses inorganic phosphate. What else does it utilize? GDP. Okay. So because we make, what do we make from that? GDP. GTP. Okay. And I think this is the first time that we've seen GTP made directly on something. Okay. So yes. Okay. And we get our coenzyme A back, so that way coenzyme A can be shuttled to either go back into the cycle here or go on to build fats if you wanted to build fats. Usually if this is going through, you wouldn't be wanting to build fats because you do this under when you need energy. Okay, but now the $25 million question. Not really. $25 million hypothetical dollar question. What class or classes of enzymes could this be, knowing what we know? What's one of the possibilities? It'd be kinase, if we were phosphorylating something. Mm-hmm. What else could it be? Along the same line. A what? I didn't hear you. Oh. What are some of the other possibilities? A what?
It has inorganic phosphate.
It's inorganic phosphate. So yeah, maybe a phosphorylase. This one, I mean, it does utilize GTP, and so we could say a kinase. What else can we call it?
Transferase.
Okay, and what would it be transferring?
Acetyl-CoA.
Well, not acetyl-CoA, but coenzyme A. If it's going backwards, you're getting. Now, if we start putting those together, you're going to get really, really close. This, by the way, is actually named for the. It doesn't make sense to me. It's named for the reverse, which doesn't happen in people. But so I'm just going to let you know that. But there is another class of enzymes that you haven't mentioned that this could possibly be, if we're going backwards. Synthetase. And what would we be synthesizing? Succinyl-CoA, and this is succinyl-CoA synthetase. I don't know historically why they named it for the reverse. It's one of those few times, sort of like,
So,
it's just one of those times that it's named for the reverse. It makes sense going backwards. It would be a succinyl-CoA synthetase. I don't know why you've got because it's using GTP for energy. You make succinyl-CoA.
So hypothetically speaking, if we were given something similar to this, it wasn't that exact, those exact molecules, well, how would we go about naming that? Like,
Usually what I do is I ask you for your rationale
okay.
to explain it. And so that's why if you had, there are multiple ways to explain it. Mm-hmm. And just like we've already seen it once before, with remember how in plants and animals, the carboxy kinase peptidase, sometimes they're, depending on who discovered it in the species or the classification system that's used, they do have two different names. Just for that reason. This is succinyl-CoA synthetase. This is the first time you get energy directly from citric acid cycle. All the other times you've made NADH can go on, but we've never made something energetic.
What exactly makes these checkpoints? Because this one's not reversible, but it's not a checkpoint.
It's not. So just because checkpoints are not always due to reversibility. It's due to the fact that they can be allosterically controlled, typically, or be controlled genetically or controlled with one of those methods. Usually what we're talking about is allosterically controlled, or covalently modified. Okay. Many times they are relatively energetically unfavorable going backwards, but not necessarily. Mm-hmm.
So it's literally like thinking like a checkpoint, like a gate where you can shut, close it.
Exactly.
So
this one doesn't. What this would way to read this is when you make succinyl-CoA, it's going to go on. It's not the determining factor. It's going to go on to make succinate. If it starts to get backed up, the way that it does help control it isn't at this enzyme level. It goes all the way to citric synthase. And citric synthase has a binding site for this, and so therefore it blocks it. Okay. We've actually seen this one before. Okay. The way, because usually we don't utilize GTP specifically for energy. We utilize ATP more often than GTP. And so we have this nucleoside diphosphate kinase, we've seen it multiple times before, and it's a way to get ATP from that GTP, where literally just the two NTPs, that's why it's got a generic name, just swaps the terminal phosphate. Okay, so that's why sometimes when they draw out the citric acid cycle, it'll look like this. Oh, that's fine. They'll have like the step 0. Ooh. And then they have this little thing here where we have that's probably not the best way. I did it in the wrong direction, but that's okay. Hopefully you get the idea. GDP to GTP plus ADP plus ATP. And so then what happens is we get the ATP, but it's not directly a part of the citric acid cycle. It's one step removed. You get technically you get GTP and not. And that enzyme right there that I just drew to the side was that nucleoside diphosphate kinase. This is a little FYI. All right. 6 and 7. I don't know why I put them on the same. Okay. I'm not sure why I put them on the same. But once again, I want to give you a chance to try to figure out what's going on, naming them based off of what you know and all that kind of stuff. Now, I don't know if I told you or not. They're not including step 0. There are only 8 steps to citric acid cycle, so we're almost finished. I've forgotten by Calcis to show you some of the videos, or a video or two that people have made in the past. I don't have a good one on citric acid cycle. That's in English. I got one in Spanish. I used to have one in Spanish. It's really fun to hear, speak Spanish. And I I think it's still chapter 20 in the book. It talks about FADH2 and making 1.5 ATP. So I think I did update that. We don't cover, due to time constraints, we will not cover the plant chapter. Since almost everybody in here is geared towards biomedical sciences, we just usually don't have time. I'd much rather go further into like looking at the hormones and all that kind of stuff than spending chapter talking about plant biochemistry. Okay, so at the very top on step 6, we have succinate becoming fumarate at the expense of an FAD becoming an FADH2. Please note it's FAD, not NAD. Also, please note FAD is truly FAD, but NAD is NAD+. That's just a little side note. Okay, so what class or classes of enzymes could this be? And it is a dehydrogenase, and if you were going to name it, what would make sense to name it? And that is true. This one is, and I'm going to abbreviate because I don't have that much room. But yes, it's succinate dehydrogenase. Step 7 is an important step. Before I forget, I was about to forget, and I don't know that your book tells you this. Step 6 is the step that is located at the inner membrane of the mitochondria. It's the part that's going to, it's also complex 2 in the electron transport chain. It's the part that's the common junction point between those two metabolic pathways, for lack of a better word. Okay, so step 7, if hydration of fumarate becomes L-malate, and it's fine with me if you just call it malate. That's the L versus the D, just let you know the stereochemistry for the sugar. We've actually seen malate before. We have. I don't know if you remember it or not, but we saw it as part of gluconeogenesis. Malate's important. It's another one that's commonly used in multiple metabolic pathways. This is another one that's a little bit more tricky in naming. Does anyone know what enzyme this one is? It's got a common name. That's the reason why I say it. It makes sense when you hear the common name, but it's fumarase. Yes. This one's fumarase. Which I don't know. When my Spanish course kicks in, fumarase, it makes me think smoking. I have no earthly idea why. I mean, I know why. I have no earthly idea why this is called fumarase. Or fumarate. They don't want to call it fumarase. If it has anything to do from the common word, because, well, if you speak Spanish, you know what I mean. Fumar, it's to smoke.
Whenever you
Please don't smoke.
Step 6 is located in the inner mitochondrial membrane. You also said it was a second point or something.
No, I call it a junction point for the electron transport chain. I don't know that's the best term for it. No, junction point, like in the subway system, you have different lines that have meet. It's kind of like that. It's part of complex 2 of the electron transport chain. Electron transport chain is 4 complexes. There. So this is something that try to keep in your mind. We'll see it again in the future. By the way, it's the reason why you only get 1.5 ATPs instead of 2.5 for FADH2. Okay. Now we've come to the end of our road for the citric acid cycle, at least. This is step 8. We go from malate to oxaloacetate. We get another NADH out of it. We've actually seen this enzyme before. Does anyone remember where did we see this enzyme? I even said, in case I forget, I'm 99% sure that I said that, just like I did for complex 2. Where did we see this enzyme before? We get malate from oxaloacetate.
Getting it out of the mitochondria.
Exactly. Mm-hmm. This was a part of gluconeogenesis, not directly, because remember, oxaloacetate is a part of gluconeogenesis. It's been a long week, even though it's been a short week. But oxalic acetate do not get to, it can't transverse all the membranes from the mitochondria, right? And so since the gluconeogenesis has to be able to go between the mitochondrial matrix and the cytosol, it utilizes this enzyme. Please note, the way that it's written right here for the citric acid cycle, they show it as being irreversible in a sense, but overall it is reversible because this is how, that's why this is not a checkpoint, because this is how we can effectively get NADH across the membrane as well, because NADH cannot cross between the mitochondria membranes and the cytosol. So this is one way we can get the electron equivalents. There's a cytosolic form of this enzyme, and then there's the mitochondrial form of this enzyme. What enzyme is this? What class of enzymes can it be? It's a dehydrogenase, and if you're going to name it, you would name it malate or malate dehydrogenase, and that's what it is. Okay. Like I said, it's one that we've seen before. Again, oxaloacetate, maybe you can see it now, you can picture it. It's the alpha keto acid of aspartate. So, if you add everything up, you get per round of the citric acid cycle. So for every one pyruvate, you get one GTP, which can go on to make one ATP. That's why some books call it an ATP. In reality, it's a GTP directly. You get one FADH2. You get 4 NADHs. However, one of those came from step 0, this little asterisk. And you kick off 3 carbon dioxides. I wanted to point out the carbon dioxides because I think that we stated this, I don't remember if it was this semester or last semester, because they started blending together. Carbon dioxide is the most oxidized form of carbon. That means you have literally been able to abstract all of the electronic potential, electronic power out of that carbon. Okay. And so it's not good to us anymore. This is one of the reasons why it's a byproduct, a waste product. We want to exhale it out. Yes, there are some enzymes that do carboxylation, but that's minor. Okay, so that's why we used all the energy we could. We used it, kicking it to the curb, so to speak. Does it sound like we're on Springer? Whoops, I did not mean to do that. This is just a little table to show the 8 steps. Please note it does not have, it's from the old book, it doesn't have step 0, pyruvate dehydrogenase complex. A lot of this I've already covered as we went along. The 3 control points or checkpoints, there are 3 within the cycle. Please don't memorize just the numbers, because one tricky thing that I've done before is on exams or quizzes. Instead of having it where step 0, 1's at the top, I turn it, turn the cycle, and when people just memorize the numbers, they get it wrong, okay? So that's why you kind of have to understand the theory, the philosophy, or the reasoning behind it, I should say. But it's citric synthase, isocitric dehydrogenase, and alpha-ketoglutarate dehydrogenase complex, and then one outside the cycle, which is pyruvate dehydrogenase complex. I've got the word complex in all. If you've got the word complex, it's fine too. I would know what you're talking about. Okay. All right. Are there any questions so far? Citric acid cycle, hopefully by now, it's starting to make a little bit more sense. You're getting a feel for the enzyme. There's the condensase and fumarase for the hard ones. The one that's named for it's backwards, but the synthetase, because it uses GTP. I like this little picture here because it just summarizes and shows everything. Shows you the relative locations where stuff occurs. So you can see the overall, the big scheme of things. They don't have the enzyme names in there, but you could even probably use this to kind of quiz yourself on like the enzyme names and functionalities and the fact that where the ATP would go in, or how do you get the ATP, like where's the GTP and all that kind of stuff. Okay. This, I just want to hammer it in. Right? Hopefully this makes sense. If the cell, I'm anthropomorphizing here, is resting. So So therefore, you do not need any more energy, right? Your ATP tends to be high, ADP would be low, so that ratio is going to be high overall. Your NADH will be high, your NAD plus will be low, so that ratio also will be high. Whereas if your cell is active, the converse is going to be true. And so we are very limited in the actual total amount of the NAD that we have around and the ATP, ADP that we have around. There's only so much, right? So, especially the NAD and ADH, we're really, really limited. And so it's that ratio that's so important. Okay. All right. Oh, this is where we get to, this is the fun part. I get excited. I really like this figure, this kind of thing. They want you to be able, some of you already know, but be able to start to put the pieces together. Okay, this is to show you essentially what we know so far, with the exception that we haven't covered fatty acids yet. But it's showing you like how the pieces start to fit together. Remember that one picture of the metabolic cell? Remember there's all those little pathways and all those junction points, and it just really is overwhelming. Whereas if you look at this, it's just a little bit more simplistic, and so it just has a couple of them. But we've already covered a lot of this. So just to review, the carbohydrates. What's an example of the carbohydrate? Glucose. That's the simplest, but we've also covered things like sucrose and fructose and all that. Those, you break them down, you get pyruvate. Pyruvate, using pyruvate dehydrogenase complex, will go on to make acetyl-CoA. Acetyl-CoA then is utilized to make a lot of these different metabolic intermediates. Okay. Likewise, I've already told you the breakdown of fatty acids. We're going to find out later on. It makes acetyl-CoA to enter in the citric acid cycle. We've talked about proteins. What class of enzymes break proteins down to amino acids? Proteases. Okay, you break it down to your specific amino acids, and we're going to find out, we actually already know some of these, but these amino acids can then also be catabolized to get energy. Okay, so you can use it. The amino acids are the only, or proteins are the only one of those classes that we can't store for long-term storage, right? You can store some glycogen, carbohydrates. You definitely store a lot of store, myself included, way too much fat, right? The fatty acids for energy. But we don't store proteins. There's no long-term storage for that. In particular, yes, you can build up muscle, but that's one of the reasons why your muscles can decrease relatively quickly as well, because of the fact that it will catabolize that amino acids. So for an example, then we break down the proteins, we get alanine. Alanine can be converted to pyruvate using what enzyme? Alanine aminotransferase. Okay. Then we could have aspartate. Aspartate can break down and turn into what? Which of these? Aspartate. Oxaloacetate. And what enzyme, what specific enzyme makes that does that? So think about if it's alanine aminotransferase that goes from alanine to pyruvate, what aminotransferase is going to go from aspartate to oxaloacetate? Aspartate aminotransferase. Okay. We could have, and we'll find out asparagine actually can also go that way. Asparagine can become aspartate. Aspartate can become. Right. So we have glutamate. We've already covered glutamate. Can become which of these? Alpha ketoglutarate. What class of enzymes can do it? It's an aminotransferase. This is a little asterisk. Keep this in your mind. You'll probably forget it. All aminotransferases do that. Alpha ketoglutarate and glutamate are involved in every single aminotransferase. Okay, so there's not one called glutamate aminotransferase. It's a little side note. Okay. We'll cover that whenever we start talking about nitrogen. How do you get rid of the nitrogen and glutamate? It's kind of a stickler. What? Well, it's hard. Okay, and then we'll cover this. So that's why we can see how it's used in catabolism. So if you prefer seeing a cartoon look, here we go. Give me a moment to take in the beauty of the mitochondria and God's creation. Here. Some of this we already know. We've already covered. Some of it we will cover throughout the semester.
So this is not to scale, right?
This is not to scale. No. All right. Does anyone want to hypothesize, knowing what you know now? So we have this alanine becomes pyruvate. So where they put a TA, what is that? It's aminotransferase. The old name, or like a common name for aminotransferases, were called transaminases, which sounds like a hot rod car. But so that's why that means it's TA for transaminase. It's transaminase reaction. But it's aminotransferase, and it's alanine aminotransferase. Okay. What we'll find out. Oh, well, this is just glycolysis. We've already done that one. We won't cover all of these. We cover some of them specifically. And as we mentioned here, asparagine becomes aspartate, aspartate becomes oxaloacetate, oxaloacetate becomes malate, malate can cross over, and then we just get the reverse, and that's how you can get it through citric acid cycle. Over here, we have some of the more complicated ones become acetyl-CoA, and then transfer into, and so on and so forth. Okay, glutamine, I don't know why they did this, but glutamine can become glutamate, glutamate can become alpha-ketoglutarate, which goes on in as well. Okay. All right. Here we actually already gave you answers. So transamination is the old name. It sounds really cool though. It's aminotransferase. And once again, specifically, this one would be aspartate aminotransferase. This one is just aminotransferase. It'll make more sense once we get to it. Because your aminotransferases are reversible. So at all points in time, you have to have something that accepts the amino group. So you can imagine if you're breaking aspartate down to get oxaloacetate, the nitrogen that it's being transferred to is alpha-ketoglutarate, so you make glutamate. Whereas, let's say that we want to go from oxaloacetate to aspartate. What happens is oxaloacetate takes a nitrogen from glutamate, so that way you make aspartate and you get alpha-ketoglutarate out of it. Same thing with alanine. So this alpha-ketoglutarate and glutamate is always they're called ping pong reactions, so they go back and forth, and it's always one of the partners, one set of partners in the doubles reaction. Yep.
So if you had more oxygen, it would go more to the ketoglutarate.
No, it's not just based off of the oxygen. They're showing it's reversible. It's just to show you some examples. It's going to depend on lots of different things. If you need, let's say that we need aspartate, and this is the way that we're going to make it, then we're going to take it away from glutamate to make aspartate. If you need glutamate, one way to make glutamate is to get it from alpha-ketoglutarate, and we can literally take the nitrogen from any of the other amino acids. Okay. So trust me, we go over lots of examples of it throughout the semester. We see aminotransferases over and over and over and over again. You have to be able to replace the intermediates, if you take it away, and that's where some of the suggested problems are similar to that, to where, and there are a lot of medical conditions associated with this, that you can imagine that there are ways, and sometimes they're called shunts, ways to get around parts of it, because if you take something out of the citric acid cycle, you've got to be able to replace it. As we've already seen, there are multiple ways to feed back into it. Okay, and so this is just one of the ways. This is pyruvate carboxylase. Just to show you from before. Okay. So that was another example for anabolism. Remember, catabolism is breaking down, anabolism is building it up. This one is a little cartoony way, or one of the cartoony ways of showing that same thing. Okay. Please note, this isn't my favorite one because it doesn't show you, it shows you things that we can't do. Okay. And so that's why we don't go this way, we go this way, but we, as in mammals, this is the way that we go for anabolism. But there are other animals there are other organisms that can go about different ways as well. Okay, but this is just to show you that we can go ahead and feed back into it to citric acid cycle. And this is the not drawn to scale mitochondria one. Okay, which should look really familiar in the sense that essentially it's gluconeogenesis. We saw this. We just added in the circle part of the citric acid cycle to show how the citric acid cycle can actually help with gluconeogenesis. If we don't need the energy, then we can build it up going backwards to store the sugar. Are there any questions so far? All right. So we've tied it together to proteins and we've tied it together to carbohydrates. This is how it's also involved with lipids. So lipids in particular, we're talking about acetyl-CoA. Acetyl-CoA and lipid anabolism is in the cytosol. So if you, and you may want to do this, like have, you know how I say you have like a page where you're putting in all your enzyme names, enzyme family names, which I suggest that you do that. You may also have one where you a cell that you start to put in like where does certain things go. We had glycolysis occurring in the cytosol. We had citric acid cycle occurring primarily in the matrix, with the exception of the one little crossover into the mitochondrial inner membrane. We have gluconeogenesis, which goes through both. But we have lipid anabolism, which we will cover in further detail later on. But it's in the cytosol. Okay. Which does anyone know where this lipid? Where's the opposite of lipid anabolism? So which is beta oxidation. Where does that occur? It's not in cytosol. It's one way that we can keep it compartmentalized. And so that's just it. Keep in mind. Okay. So we have to have some way to get around it. Oh, you can ignore this one. I've already fixed it. I forgot to take that point out. Oh, it's because now it's a PDF. That's why I couldn't do that. Okay. Because originally it had a typo in it. And it was a picture, so I couldn't go in and fix it myself. But I found it right. Okay. So acetyl-CoA is just like what we've been talking about with NADH and oxaloacetate. It has problems sometimes getting from one place to the other. And so we have to have an indirect transfer to do that. And so this is one way to do this. And on your own you can go through. But it utilizes citrate. Okay. We'll go into more detail later in the semester when we go over lipids. I just wanted to show you that it wasn't limited only to proteins and carbohydrates. So this is how, and this is also another way that you can effectively get an NADPH. Remember we also talked about potential phosphate pathway. This is like a backup to get NADPH. So, yeah, no. So I don't know if there's anything else that you wanted to talk about. Are there any questions? Because I got a little cartoon to show all this at this moment. And then you see it word-wise first. Okay. So here is the not drawn to scale mitochondria depicting this. There's a lot going on, but it's letting you know like how the anabolism is working and how we can build other things from it as well. Some of the cool things I just want to point out. I mean, we've already seen this in gluconeogenesis. We've already talked about, to make proteins with various amino acids. Okay, but I want to point out this is one of the ways where we can go ahead and make the fatty acid-derived stuff. But we can also make porphyrins right here. Okay. And one of our porphyrins, we cover this later on. You can almost turn porphyrin ring into like a devotional. What are the porphyrins? We've seen it once or twice so far, primarily last semester actually. What's a porphyrin ring? What's it in or make up? One of the diseases, there's lots of diseases associated with it. One is called porphyria. Some people equate to vampirism.
Is it similar to an aquaportin?
No. Porphyrins are make up the heme hemes for hemoglobin and other cytochromes and stuff like that. And so, which are huge. These are these huge, relatively planar looking molecules. They are made up from really, really tiny things. And so the amino acid that's the building block for it is glycine. The simplest amino acid can actually be used to build one of the most complex things. And so that's why I say you can actually convert that really into a devotional almost. Okay. I've got one or two slides that talk a little bit about plants, but then the medical implication of this, I want to say, because I like to go over some of the medical diseases associated with the citric acid cycle in greater detail. So I want to save that for Monday. And so are there any questions? No, we only have a couple slides left. Are there any questions? That way we can go into, spend more time talking about why some of these diseases, I don't want to say make sense, but why they make sense logically, metabolically. Any questions? Okay.